Thank you for joining us for the EBI Women's in STEM Seminar Series. Our guest speaker today is Karine Gibbs, who is an associate professor here at Berkeley that specializes in social behaviors of tiny organisms. In 2020, Karine was recognized by Cell Press as one of the top inspiring Black scientists in America. Hello, Karine. Thank you so much for joining us today.
Hello, Yi. Thank you for inviting me to join you, and thank you to you and the EBI for this interview. I'm looking forward to our conversation.
Yes, great to meet you. Today, we are going to really try to understand the basic topics of what Karine is specialized in and how it has an impact on our understanding of bacteria. And what we're going to touch on, we're also going to touch on her career and how she's gotten so far. So first, let's talk about some background on the science that is involved in your research. I think a lot of us understand what social behavior is in a human means, but when we talk about social behavior of a cell or a bacteria, what does it mean and why should we care?
So let's take a step back. When you look at the pink slime that's on your shower curtain, or you look at a mass of bacteria on a tube that clots it, whether it's a catheter or an industrial tube, those things, those communities are called bacterial biofilms. And several of my colleagues in science showed years ago that inside of those communities, bacteria are talking to each other. They're using small molecules to say who they are and to drive changes in the expression of genes necessary to control behavior. So the term social microbiology or sociomicrobiology was first created by Dr. Pete Greenberg and his colleagues in the 90s. And it's to talk about how do individual bacteria work together to make a colony or make a community. And now, for example, the gut is an area of community behavior that we study. So, for example, the gut microbiome, or the bacteria that live in your teeth, or the bacterial groups that live on the soil, so on a root nodule. All of those situations require bacteria to interact with each other, to coordinate, and now we understand also communicate. So my group in general is really interested in how do siblings talk to each other, and specifically, how do they share markers of who they are? How are they different than either their cousins or something really foreign? And then how does the communication of that identity work in terms of the mechanisms of cell-to-cell signaling, the changing of individual cell behavior, and then how does those interactions on the single cell level lead to group interactions? And so specifically, my lab studies collective migration. How do bacteria move over centimeter-wide distances as a whole community when a single individual can only go tens of microns, so 100-fold smaller? And we studied the role of identity signaling and communication in that.
Wow, that's really cool. So I guess a lot of people would have the question is, what areas does this research impact? You mentioned that you study like bacteria in the soil or in the shower.
That's right. So I have a confession. I am a traditional basic biologist. And what I mean by that is that my job is to create new knowledge. So really to expand what we understand about biology. My graduate advisor, Dr. Julia Fitterio, used to describe it as if you imagine the light from one of those traditional um, light posts, there's the bright light in the center, and then there's areas that are a little bit gray right before you get to the darkness. I think that the strength of my group is that we sit in that gray area and we cast out into the darkness and we try and drag the light over to where we cast to. And so when you're asking the question of what are the impacts of what we do and how it kind of matters for your life, on the most fundamental level, we're asking, how do bacteria signal with each other, and how do those signals drive the formation of structural units, these communities that have impacts on larger organisms. So whether the plants, animals, etc. So what we do is that we generate new ways of manipulating the genetics of organisms. I happen to study an organism called Proteus mirabilis. It's a fancy word to say an organism that is able to change shape rapidly and move. It is an organism that lives in your gut, and then it uses its collective migration, that social behavior, to cause disease when it migrates to your bladder. This organism, before we started working, didn't have good genetics in it. And so one of the things we've done is build new tools and adapt old tools to be able to manipulate an organism where others have not not easily done it before. We also do a lot of technology development for microscopy, meaning we come up with new ways of analyzing bacterial shapes and then analyzing groups of bacteria, and we make those available for the community. We discover new targets for antimicrobials, so ways to stop the transition from something in your gut to something in your bladder. What we're hoping to do as I've moved here to Berkeley is understand more with soil microbes who do the same thing. So organisms that live in the soil that also have this collective migration that's important for their behavior and interaction with, in this case, as an organism that kills pests, trying to understand how to manipulate that using the tools that we have in Perdia. Um, I think, though, most fundamentally, I think that our work helps us understand collectivity in general. So where we see other collective migration is in like birds flocking or fish swimming. And we have some ideas of how interactions between individual birds and individual fish can amplify. But those organisms, it's hard to change the genetics of them. So it's hard to change the genes. And it's really hard to change how they talk to each other. Um, but with bacteria, we can do that. And then we have the added benefit of being able to make mutations and see how those mutations can lead to new adaptations and changes. So evolution. And so, if you think about where I'd love to see our work go in 10 years, like that big impact, I would like us to really have built a system where we can manipulate the controls for collective migration. And hopefully from learning what those features are, inform collective behaviors of larger organisms like animals.
Mm.
But that's outside the scope of EBI.
No, no, no. That's really cool. So I guess it sounds like your research area is quite new. How long has it been around?
That's right. So social behaviors of microbes has been around since the 1970s and a little earlier. And it's actually started off by asking, why do squid glow? And so the Hawaiian bobtail squid has a light organ. Inside of it, it has bacteria that, when they're at high density, they make a small molecule that they eat, that they share with each other. It causes light to be produced. That was the basis of form sensing. And that has actually led to the formation of understanding the role of form sensing in diseases like cystic fibrosis, et cetera, has led to new companies and small molecules. The work that I do in collective migration and Proteus and this kind of kin identity and the role of it, that the first inklings of it was in the 1940s, but quite frankly, it's only in the last 10 years or so that we've actually been able to understand the genes that control it, understand that it's different than traditional quantum sensing, that the cells actually have to touch each other and jam information in. And then the work with collective migration. That was actually a different part of microbiology that's been going on for a while. And we actually still don't understand it at all. And so collectivity itself has only been really, on a molecular level studied in the last 5 years. And collective migration, we are, oh, wow, we are in the early days, really early days. I don't want to put numbers on it, but let's just say it is hot and heavy. We are just now starting. So if you go on into journals, you're just now starting to see some really cool work coming out.
That's really amazing. So, how would you encourage students to study this in the future? Or like do basic research, maybe?
There are many different ways of approaching science. And as a student, you need to understand and figure out what areas you thrive in. So whether that's direct applications, so those are my colleagues who are working on systems where we really understand what's going on. I like to think of E. coli as that. And you can build it to build new technologies in either bioengineering or similar. Or if you're someone who likes to discover new mechanisms, so understanding, finding organisms that do some new feature in in the environment, like bioremediation or plastic degradation. Or are you someone who likes to do work at the edge of science, like I do, in generating new knowledge, where we don't know what we're working on is going to lead to direct application tomorrow, but we know that we're building the fundamentals so that other people can then build their research on. Or whether you're someone who is much more interested in generating the interest or financing of science. There's space for all of that. I think specifically for our type of research, the generation of new knowledge, nowadays, I'll say very practically, you need to understand genetics, computer science, and mathematics. They are the bread and butter. It's integrating multiple disciplines together because often the area that we see new knowledge happen in is when you see the junction or integration of different fields. So really becoming um, secure in your knowledge of that. Also, giving yourself the grace and space that failure happens. If you're throwing things out into the darkness, not all of them are going to be spaces where you can actually pull back. Sometimes the line's going to break and you have to do it again. And then the last thing I'll say is that I think it's important not just to think about what technology can be done right now with what you're building, but try and think about what technologies could be built if you get that answer, right? So go one step further away. And so one of the things that you and I have talked about previously is that in my group, one of the fun things in this process is that we've discovered new enzymes. We've discovered new proteins that are able to degrade DNA in ways that weren't known before. And so those can be now adapted for direct application. So for whether or not in, degrading DNA for treatments or using it for others in basic science.
That's great. So I guess the next few questions is going to be about your career as a researcher, which I'm sure your audience is going to be very curious about. How did you become a associate professor at UC Berkeley? What was the process like for you?
So I'll give you a brief history. I was an undergraduate at Harvard, then I went to graduate school directly. I went to Stanford and did my PhD there, and afterwards I did my postdoc and with Dr. Pete Greenberg at the University of Washington. And I went there because I wanted to work with Pete. He is one of the fathers of quorum sensing, and I had always been interested in how bacteria talk to each other and why they have these community behaviors. And when it says there, said, actually, have you heard about this organism over here? Look, they can move. And when I looked at them, you would see individual cells would just go a few microns, so 10, in distance, but the whole colony could cover a full centimeter in under 3 hours. That's a lot. That's 100-fold greater speed distance. And so that project and that question of how do they do this and what's the role of talking to each other in it, since it wasn't traditional quorum sensing, that is what became the basis of my research as an assistant professor at Harvard. As I said before, we had to build the system up, and so we spent a lot of years doing that. And then I had the opportunity to move here to Berkeley, where one of the attractions to me was being in a department where there were other faculty, actually most of the other faculty, study how organisms interact with each other, whether it's bacteria in plants or bacteria in bacteria. And I also love the idea that the mission of the Vassar College of Natural Resources, is to give back to its community. Um, and so it's forced me to think about my science, not just in the most basic way, but also what can we do that has direct applications? For example, the questions you're asking earlier.
Being a minority in STEM and in industry can be difficult, and some even more difficult than others. For example, there are only 2% of Black women currently in an occupation in STEM. However, despite these stats, there are extremely talented people, including yourself, in this field. What made you decide to study in STEM? And were you ever interested in choosing another field?
So I study STEM. I specifically study biology because I love science. I've always loved science. I love the questions you can ask. Um, it is challenging to be a Black woman in academic science, but I think it's challenging to be a Black woman in many aspects of our society. Um, I will say that the question of why am I still in academia is that I'm stubborn and I really love what I do. I love working with students. I think that I was saying to you earlier that I am so impressed at the younger generations. And I can see how society is moving forward and changing. And I love that you have a passion for making sure that you take care of yourselves in addition to taking care of the work that you do, which was not always the case when I was training. Those were not the messages I received. Um, I also think that's really important that as someone who is a mid-career woman, Black woman in science, both as a Black person, as a female, as a first generation, my family I was born in Jamaica, but I was mainly raised here in the States, um, that I have a responsibility to try and make it easier for everyone who's younger than me, because there are generations of people above me who did the same. And that's part of my responsibility. I have the privilege of being here, but that means I also have the responsibility to be hopeful, to be optimistic, and to really shield and folks behind me while changing what's coming, right? So pushing the barriers above me while also smoothing the path behind. Um, and so that's what I try and do. I'd say the place where I questioned the most was between graduate school and postdoc. I very reluctantly did my postdoc, and I do not regret that in the least. I loved working with Pete.
Mhm.
He was exactly the postdoc mentor I needed at that time. But coming out of graduate school in the Bay Area, at Stanford, at a time when our salaries were our stipends were much lower than they are now, it was really hard to choose to stay in a discipline where it was such a struggle to financially make it through graduate school. As I said, I love discovering new things. I really what I thrive on is the creation of knowledge.
Oh, that sounds like an amazing journey. So in the essence of time, one last question. What would be the one piece of guidance you would wanted to tell your younger self or other young people in the field of STEM? Um,
Yeah. So I'll make it two pieces of advice, but it's the same thing. They're two pieces of the same coin. One is that I think it's really important to choose yourself and to choose to thrive. There's a lot of messaging to suggest that science should be the be all and end all, and we are in a marathon in life, and you only get to live it once. So this doesn't mean go wild and crazy, but really ask yourself, what is the situation? What are the questions? What is the research you'll do that will feed your soul? And I think, lean into that. Clearly do it in a safe way, but that's what I would recommend. And the number one thing that I would say to my younger self is that at some point you have to decide to stop caring about other people's opinions of you. And I say this especially as a minority in science. I say this especially as a woman, where I think a lot of women, myself especially, was really sensitive to other people's comments. And that's not to say to close yourself off from criticism. I think criticism is good. Trust me, I get a lot of it. um, but I think that at the end of the day, science is a creative art. And you have to decide that when you put something out there, before you put it out there, you need to settle how you feel, the good, the bad, the in between. And then once that you've settled on it, let it go because everyone's going to have something to say. But really, at the end of the day, when you're on your deathbed, the only thing that really matters is how you felt about your presentation to the world, your addition. So choose yourself.
Thank you so much for such a great conversation. It was really, truly amazing to see a woman that is so accomplished in your career so early on. You're a great example for our future women looking to enter STEM field. I, alongside with EBI, would like to thank you for your time and wisdom. We look forward to seeing what comes next of you, and we wish you the best of luck in your future ventures.
Thank you. And thank you so much for having me. And thank you for taking the time, everyone in the internet world, for listening.
Thank you all for watching. Please make sure to like and subscribe, as well as click the bell to be notified for our next interview. Our socials are linked here as well as on our website. If you're interested in climate change, renewable energy, biosciences, and all that comes with that, and also looking to see what career path you would like to take if you're interested in that, we highly recommend checking our socials and website out.